Hi and welcome back to Free Science Lessons. By the end of this video, you should be able to describe how to use a bubble Plotometer to measure the rate of water uptake in a plant. You should then be able to describe the advantages of using a Mass Potometer. In the last few videos, we've been looking at how water moves through a plant. We saw that water vapour evaporates from the moist surfaces of cells inside the leaf, and if the stomata are open, then this water vapour can diffuse out of the plant. Scientists call this process transpiration, and the effect of this is to draw water up the xylem vessels. Now we can measure the rate of water uptake into a plant by using a bubble Plotometer, and I'm showing you a bubble Plotometer here. A bubble Plotometer consists of a fine capillary tube which is filled with water. The tube is connected to a plant which is being cut at the stem. The tube is also connected to a syringe filled with water. And finally, we use a needle to place a small air bubble at the end of the capillary tube. As water evaporates from the leaves of the plant, water is drawn into the stem. This causes the air bubble to move towards the plant like this. By measuring how far the air bubble moves in a given time, we can calculate the rate of water uptake into the plant. We can then see how the rate of water uptake changes if we change the conditions. For example, we could carry out the experiment under different light intensities, or we could investigate the effect of wind by using a fan. In between experiments, we can reset the position of the air bubble by adding more water from the syringe. Now you need to bear in mind that this potometer only measures water uptake into the plant, and not all of this water will take part in transpiration. For example, a small amount of the water taken in will be a reactant in photosynthesis. However, the vast majority of water taken in by the plant will take part in transpiration. Okay, now there are a few things you need to consider when setting up a bubble Plotometer. Firstly, when we take our cutting from the parent plant, air will be sucked into the xylem vessels, and these air gaps would prevent water from being taken up the stem. So to correct for this, we now place the cut stem of our plant into water and cut off the last 1 centimetre. Water will now flow into the xylem and we'll avoid any air gaps. We then need to place the Plotometer under the water and insert the cut end, again avoiding any air gaps. It's important when doing this that we handle the plant carefully. We don't want to damage the plant, and we want to avoid getting water onto the underside of the leaves, where most of the stomata are found. Now it's important that the Plotometer is fully sealed, so it's a good idea to smear some petroleum jelly around the connection between the stem and the tube. We also need to allow the plant to adapt to its surroundings for 10 minutes before starting experiments. Okay, now there is another type of Plotometer. This is called a Mass Potometer. In this case, we place our plant in its pot on a balance. As the plant loses water through transpiration, the total mass decreases. Now with this Plotometer, we have to prevent evaporation of water from the soil. Otherwise, this would contribute to mass loss, giving a false reading for transpiration. And we do this by covering the soil with plastic wrap. Now, there are two advantages to the Mass Potometer. Firstly, this directly measures the rate of transpiration, rather than the rate of water uptake. Secondly, this is much less disruptive to the plant, as it does not involve cutting the stem. Okay, so hopefully now you can describe how to use a Plotometer to measure water uptake into a plant.